Hi, I'm Jacqueline, and I'm here to talk about today DNA replication. So first, as you can see here pictured, is a DNA bubble. This is where the two DNA strands begin to separate. Here, where I'm about to highlight, this is a replication fork. This is where the bubble is split into Y shapes. Helicases, which is an enzyme, this enzyme comes in right here into the replication fork where I was just speaking of, and it snips the H bonds, or it untwists the double helix at the replication forks in order for the DNA to begin to replicate. Inside the replication bubble here, there are something called leading and lagging strands. So first I'm going to draw the leading strands right here. Now, it's very important to note that the leading strands are always heading towards the replication forks because they're always trying to make the bubble bigger and bigger so they can eventually become their own separate DNA strands for replication. Now I'm drawing in the lagging strands, which are behind it, and as you can see, they're going away from the replication forks. Now, these little fragments of the lagging strands are called Okazaki fragments. These little things right here. And that's very important to note later for a different part of the process. Just a little side note here. The DNA strand only elongates from the 5' to the 3' direction. Now, speaking of primer is behind all of these strands here. We have primer here, 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 and here again. Now, the primer is the initial nucleotide strand in a short RNA primer. Now, primase is like a little helper that can start an RNA chain from scratch and adds RNA nucleotides using the parental DNA as a template. So it looks at it and goes, how can I add the RNA nucleotides based on what the DNA already is? Now, sometimes here, this is where different types of mutations can occur. I mean, they can occur pretty much anywhere across here, but this is kind of a commonplace. So now that occurs, our little friend called DNA polymerase 1, he's going to come in. This is how it's commonly written out in the scientific world. He's going to come in and then go right here into the nucleotides and remove them from the primer and just inject them with DNA nucleotides so it can start resembling the new DNA. Then, its sister, DNA polymerase 3, that's going to come in and add more DNA by adding the nucleotides to the 3' end of DNA, the DNA strand, or the RNA protein. Now, this next process is one of the most important proteins in this whole DNA replication process, and it's called the single-strand binding protein, or most commonly SSBP. Now, this protein keeps the single-stranded DNA in place until it can be used as a template for another structure. I kind of like to think of SSBP as kind of like a parent figure for the DNA because it makes sure everything's okay and it keeps an eye over it until it's ready to be used, because you don't want just DNA rolling around when susceptible to mutations and all different kinds of bad things. Now, finally, in our DNA replication process, we have ligase that comes in here. And he replaces the primer to join the Okazaki fragments for good. So he's going to come in here and he goes, so it can find just one giant DNA strand so it can be together forever and living in harmony. Then, last but certainly not least, topoisomerase comes in and corrects the overwinding. So it makes the DNA structure able to have that double helix like we're all familiar with. Now, I like to think of topoisomerase as kind of like an army before the king to make sure the king is safe. So it's going ahead and it's like, oh, we can't overwind, we can't do this, we can't do that. We have to make sure the DNA is safe and correct in how it's supposed to be. So I hope that you learned a little bit today about DNA replication. And yeah, thanks for listening.